Coastal Acidification Network, or SOCAN. In short, the series lays a foundation for the state of the ocean acidification science in the Southeast region. This webinar, along with all of the other webinars in the series, is intended to create a dialogue among scientists to identify what is known, what isn't, and what research in other regions of the United States can be applied to better understand ocean acidification and its impacts in the southeastern region. With awareness of and access to the research and its applications and uses, webinar participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. I'm Paula Keener. I'm a marine biologist and director of the education program with NOAA's Office of Ocean Exploration and Research, and I'm a member of the SOCAN Steering Committee, and I'll be facilitating today's session. We also have Jen Bennett-Mintz, who is Education and Outreach Coordinator for NOAA's Ocean Acidification Online, who will be organizing your questions during the webinar. During the presentation, you all will be in listen-only mode. You're welcome to type questions related to technical issues in the presentation questions tab in the GoToWebinar control panel on the right-hand side of your screen. Jen will be monitoring the incoming questions, and we will respond to them or pose them to our speaker after the webinar. We are also recording this session, and the video recording and a PDF of the presentation will be available on the SOCAN website shortly after the presentation. So we are very excited today to have Dr. Scott Noakes, a research scientist at the University of Georgia's Center for Applied Isotope Studies. Scott received his BS in Petroleum Engineering from Mississippi State University and an MS and Ph.D. from the University of Georgia in Marine Science. Scott has been involved with ocean acidification, CO2 monitoring, paleontological, and sulfuric geologic studies at Grays Reef and surrounding areas for the past 2 decades. His home department at UGA is involved with marine environmental surveys in estuarine and coastal regions and utilizes isotopic, elemental, and organic analytes. His program also houses an accelerator mass spec used in carbon dating organic-based samples such as shells and bone fragments, many of which have come from the Grays Reef area. In addition to Scott's work at UGA, he is also Director of Diving Safety and a Diving Safety Officer at the University System of Georgia Scientific Diving Program and an AAUS organizational member. So Scott, I'm going to turn the control over to you for the presentation. So welcome, Scott.
All right, thank you, Jen and Paula. And thank you for inviting me to give this webinar and to let people know what we've been doing at Grays Reef. Often I'm asked what it's like to go out and work on a buoy that's roughly 17 miles offshore, and I tell them it's a lot like getting on a ride at one of the water rides at Six Flags, where you're rocking and rolling in almost any direction, 360 degrees of motion, with the occasional wave breaking over the buoy deck. To make it even more fun, typically I have a computer sitting on my lap that you prefer not to get wet. So the thing is rocking, rolling, got waves coming over, and it's quite an experience. But if my part isn't enough fun, I usually have a couple divers working underneath, and they're having to experience all that, plus have the up and down motion and trying to undo bolts and things. So it's really quite an experience for us to go out there. And in the picture in this title page, this is what's called the ideal situation, where it's nice, flat, calm water. We don't get that at Grays Reef very often. It typically is rocking and carrying on quite a bit. So like I said, it is definitely quite an experience to be out there working on the buoy. Before I get into the operation of the buoy, I thought I'd give a quick explanation, overview of what ocean acidification is. This is a poster that Amanda Camp, a scientific illustration student of mine, did about a year ago as part of her senior thesis. She needed to research what ocean acidification was and try and put it in a way to explain it so that people could understand what's happening. Basically, throughout our normal lives we need energy, electricity for living, we produce CO2 through industry, making products. Anytime we're burning fossil fuels, we're putting carbon dioxide into the atmosphere. Now, some of that gets taken up through the plant life, our trees, grasses. It'll take up CO2 as part of their process, and then part of it, roughly 25 to 30%, goes into the ocean. So we're putting all this excess CO2 into the atmosphere. Some of it gets taken up in plant life, some of it's taken up in the water. The problem is when it gets taken up by the water, it reacts. The carbon dioxide reacts with the water, H2O, and becomes carbonic acid, thereby the term ocean acidification. Well, that carbonic acid fairly quickly breaks down into two more products, bicarbonate and a free hydrogen ion. And if you remember in your high school and college chemistry classes, the measure of pH is based on the free hydrogen ion. When you have more hydrogen ions available, the water is lower on the pH scale, and if you have less available, then it's more basic. So as we're putting the CO2 into the water, becomes carbonic acid and then breaks down where you have bicarbonate and the hydrogen ion, we're making the water move in the direction of the pH acid section of the scale. Now CO2 also reacts with water and carbonate to form more bicarbonate. And where that problem comes in is every living organism that has a shell or a skeleton is going to need calcium carbonate to build that shell or grow the skeleton. And as more of the carbonate is taken up and put into the compound of bicarbonate, there's less of that calcium carbonate available for the organisms to be able to grow. So it's basically down to chemistry, the reaction that is going on. And this poster does a very good job explaining it, showing the chemistry that interacts as the CO2 goes into the water. Now, if anyone is interested, this poster is available to download on both the Grays Reef webpage and the Ocean Acidification Program webpage. So if you want to download it and print your own, use it for educational purposes in the classroom or whatever, it's there and available. Now, Grays Reef, where the buoy is located, is about 17 miles directly east of Sapelo Island on the Georgia coast. Now, the Georgia coastline is centered up approximately in the South Atlantic Bight, which starts up around Virginia, North Carolina area, and it's this indented part of the eastern coastline forms what's called the South Atlantic Bight. So Grays Reef is a good representation of that area. Now, another part as to why would we want to put something at Grays Reef, other than it's just a convenient place in the middle of the South Atlantic Bight, is this is a very vibrant live bottom area, something we don't have much of off the coast of Georgia. But Grays Reef is home to soft and hard corals, sponges, many invertebrates. We have larger vertebrates. You can see the picture of the nurse shark hiding under the ledge. We have a lot of snapper and grouper, which are very popular with the fishing community. So it's just a very vibrant area, a place that we would like to protect and know what's going on out there. Now, unlike the coral reefs to the south of us in Florida, Grays Reef main structure is really a dolomitic sandstone. So it has a different base than the coral reefs, but it allows these organisms to attach and thrive and to create a habitat for the larger animals to live as well. Now, to give you an idea of the level of effort and organizational structure that goes into keeping this buoy active out there, it starts with NOAA. We've got NOAA as the center part. The National Data Buoy Center was nice enough to modify one of their buoys to fit the instrumentation to give us a platform to be able to operate off of. The funding for this program comes from the Ocean Acidification Program. Without that, we wouldn't be able to keep this system going. The sensors are all housed and distributed from the Pacific Marine Environmental Lab out in Seattle. They also receive the data. They are basically the storage facility for all of the data that comes off the buoys. And then without Grays Reef National Marine Sanctuary, it certainly could not happen because they are providing us with vessel and diver support to get out there and work on it. It's about a 40-mile run from Skidaway Island, where the Grays Reef office is, to get out there to the buoy. So it's not a simple task just to run out there to do some work on this buoy. And that is, as I said, it's also about 17 miles directly east of Sapelo. So it's offshore. It's not readily accessible. You do need to have vessel support in order to get out there. Now, the funding from the Ocean Acidification Program goes through SECOORA, and they are part of the IUS program. So they're part of the whole ocean monitoring system. We also have the Coast Guard. Since this buoy is also a navigational aid, the Coast Guard is in charge of recovering and deploying the buoys as they need to be replaced. So they provide, on this last one out, they provided I think it's a 225-foot vessel to go out and recover and deploy the buoys. Just the cost of that alone would be considerable for just one project to have to fund that part of vessel time. Now, the University of Georgia, where I'm housed, is where SECOORA funds us to maintain and oversee the operation of the CO2 system on the buoy. Now, this system was originally started as a grant through, hopefully I'm getting it right, PMEL, Pacific Marine Environmental Lab, back in about 2006 with Wei-Jun Kai, who has since moved to the University of Delaware. And he is still in charge of doing the discrete sampling for the quality assurance part of the system, and also underway CO2 mapping of the South Atlantic Bight. He'll also go to the buoy several times a year with his underway system and take readings, and we'll compare that with what is coming off the buoy to make sure the buoy is still putting out accurate data. And then one more into the program now is Georgia Southern University. Danny Gleason is teaming up with us at the University of Georgia to take this data to the seafloor and try and follow what is happening with the benthic community, this thriving live bottom community down there at Grays Reef, and how it will react as water chemistry changes. We get the increase of CO2, which then decreases pH. We need to find out what is going to happen to that live bottom community as this change occurs. Okay, so what we've got out there at Grays Reef, we've got your NDBC buoy, which is a standard weather buoy, and it has the typical wave height, wind speed, water and air temperature, atmospheric pressure, and that is all transmitted to NDBC through an iridium system. Now, as I said, we were able to modify the NDBC buoy to put on the CO2 system, which measures PCO2 in the air-sea interface. We have a high-resolution pH unit mounted directly under the buoy, and a water quality unit that does the standard water quality, dissolved oxygen, temperature, salinity, turbidity, and chlorophyll. Now, also at Grays Reef, we have a seafloor package, which has a PCO2 system that does the CO2 in the water plus temperature, high-resolution pH, and water quality, which does all the standard dissolved oxygen, temperature, salinity, conductivity, pressure. So we're trying to see what differences there are by measuring on the surface, and then also measuring on the seafloor where the benthic community, the live bottom, actually lives. And then all the data from the buoy is transmitted every 24 hours. This is the CO2 system end of it, which has a separate iridium system from the wave height and wind speed, goes to PMEL every 24 hours. Now the sensors on the seafloor are autonomous, and we get that data every 3 to 4 months when we rotate out instruments, bring up the old instrument, download the data, put new ones back out there. Now this is what a nice clean looking buoy looks like as it's going in the water. It doesn't stay like this for very long. As you can imagine, a buoy is sitting out there, the only thing on the water within roughly 20 miles. The birds love it. So very quickly you're going to see a layer of black slime on top of the buoy deck that's about like grease. Makes it very difficult in order to get on there and do any work. And also we get a lot of the megabalanus barnacles growing underneath, fouling the underside of the buoy. So very quickly you start getting a lot of fouling on this buoy. Now luckily in this process we have NDVC and the Coast Guard handling most of the general buoy operation, so we don't have to handle that part of it. Now as I said, we've got our CO2 system mounted on the buoy, and NDVC modified their buoy in order to put in some wells for the instrumentation. We have a battery pack on the far right, followed by a span gas for calibration purposes, and then we have the electronic package that houses a onboard LICOR CO2 analyzer plus the iridium system. And then there's also a equilibrator that traps the headspace gas, and that's what gets sampled for the actual CO2 in the water column. Then underneath we have a SAMI pH, which is a high resolution pH mounted on the buoy bridle, and a seabird that's doing the water quality. These units underneath we have to do by diver operation, where they go under and have to manually remove those, put fresh ones back on. Now there's a picture of the buoy right after it's dropped in the water. Everything was tested and operational before they turned it loose. And then a picture of the Coast Guard cutter that they use. Like I said, I think it's a 225-foot vessel to recover and deploy the new buoys. So the Pacific Marine Environmental Lab has a system of buoys. These are coastal and coral reef buoys that are doing ocean acidification monitoring. Starts all the way up in the northeast with one in the Gulf of Maine near New Hampshire. There's one at Bermuda, and then Grays Reef, of course, is representative of the South Atlantic Bight. We've got Florida and Puerto Rico, one off Mississippi, California, state of Washington, Alaska, several in Hawaii, and there's even one in Australia now. Altogether, there's roughly 20 coastal and coral reef monitoring moorings out there that are monitoring the ocean acidification parameters. All of these transmit their data every 24 hours to the PMEL facility, where they, along with groups like the University of Georgia, monitor the data that's coming in to ensure that the system is working properly. So it's actually quite a handful. They're getting all of this data that they have to watch over. Now the data is accessible from the website. PMEL has a website where you can look at the data that has come off. Every 24 hours fresh data is put on, and you can either get in a 30-day snapshot of the data, or you can click on give me all of the data starting from the beginning of this particular monitoring station to date. This one is just showing the 30-day. But you can go to this website and take a look at the snapshot that is coming off the buoy. Like I said, you can either do the 30-day or the total. You can also go to the PMEL website if you would like the actual data. The data through 2013 has gone through an extensive quality assurance effort and is available to the public in order to be able to do your own plotting and manipulation of the data. Now this is what the data looks like Starting in 2006 through 2013, you can see there's a very clear cyclical pattern that is going on out there. This is typical and is seen in a lot of the coastal sites. It is largely temperature driven. As the temperature cools down in the wintertime, the CO2 is drawn down in the water column. As it warms up, there's more CO2 available. And as you can clearly see, it doesn't take a lot of imagination that there is a very definite upward trend in the PCO2 concentration in the water and also in the atmosphere. Now, if you overlay temperature, as I was saying, there's a very strong correlation to water temperature versus the CO2 gases in the water column, that as the temperature draws down in the winter, it will pull down the CO2. In the summertime, it increases. So what is happening when, in the wintertime, when you're seeing CO2 levels below the red atmospheric CO2 concentrations? What we've got going on is a sink where atmospheric CO2 is being pulled into the water column, and it's actually creating a sink. Most people would like to believe that all the CO2 we're putting up in the atmosphere is being pulled into the ocean, but unfortunately along our coastal zones that is not always the case. During the summer months, when you're seeing our blue line, which is the seawater CO2, go above the red atmospheric line, CO2 is actually coming out of the water column. It's going back into the atmosphere, so that the mid-shelf area where Grays Reef is, those waters are actually becoming a CO2 source. CO2 is coming back out of the water into the atmosphere during that time period. So that the concept of that all this excess CO2 is all just going to go into the ocean doesn't hold true in all areas. Now, overall, the oceans are taking up about 25 to 30% of this excess CO2, but there is some leakage along the coastal zones, especially during summer months, as you have more CO2 in the water column. Now, the graph on the left is one that many people have seen. The observatory they've had at Mauna Loa in Hawaii has been monitoring atmospheric CO2 since the 50s, and they've had a constant increase in atmospheric CO2 from the very start. Now, the plot on the right is the one that I've already shown you, the CO2, air-sea CO2 at Grays Reef. Now, atmospheric CO2, as seen in Hawaii, has been increasing at roughly 0.78% per year. And at Grays Reef, we have seen roughly the same, but it's 0.789, which stands to reason that you're going to see similar atmospheric increases in the northern hemisphere, fairly well mixed as our atmosphere gets circled around the Earth, so that the CO2 is going to be relatively well mixed. Now, the point that is quite interesting is the fact that we are seeing roughly 2.4% a year increase in seawater PCO2 at Grays Reef, which is higher than the 0.78% in the atmosphere. This has created a fair amount of discussion as to what is going on, why is this additional increase of CO2 happening in the seawater when your supply of atmospheric CO2 into the seawater is actually less. So it's still kind of puzzled everyone and has created quite a bit of conversation. Now, the worldwide seawater CO2 uptake is about 0.5% per year, which is less than the atmospheric increase, and that's more of what you would expect. So that's representative of the oceans as a total sink of CO2. Now, as I mentioned earlier, we also do monitoring on the seafloor We want these instruments down there with the benthic community so that we can see exactly what they are experiencing in the way of water quality. And at this seafloor platform, these are all diver deployed, they're autonomous. We get our data when we recover the instruments. But what we've got is a wet labs water quality unit that does dissolved oxygen, temperature, conductivity, salinity, pressure, turbidity, chlorophyll. We also have a SAMI CO2 unit which can do CO2 measurements at depth. There are only a few types of CO2 instruments out there that can do this. It does the PCO2 measurement plus temperature, and then we also have a high resolution pH. This gives us the total package to be able to see what's going on down there with the benthic community. This is a plot of the data, combination data from the seafloor and from the buoy on the surface. The top two lines, the orange and blue, are showing salinity, the blue being the surface salinity and seafloor being, or orange being the seafloor salinity. What is really of interest here is the fact that we have been seeing these PCO2 spikes on the seafloor that are not represented at the surface. And this has been quite a puzzle for years. We saw it starting when we first deployed these instruments, I think in 2007. We saw these with no real explanation. This site, Grays Reef, is in the mid-shelf. There's a very long shelf that goes out there. So upwelling has really not been a prime target for what is happening. Since we do have input from the rivers on Georgia, Georgia's got about 7 major rivers that empty into this area, and we do see effect of the rivers coming out, but they should be lower salinity, so most of that effect will be closer to the surface. So this has been quite a puzzle, and it wasn't until our very first SOCAN webinar by Rick Wanikoff that he showed this slide that where he was doing some aragonite saturation measurements in the South Atlantic Bight. He did a transect off North Carolina, he did one off Georgia that went right through Grays Reef, and then one off Florida. Here's an enlargement of the one from that went through Grays Reef. I've put roughly where Grays Reef would be on this cross section. And what is happening here is you've got higher CO2 water at depth in the Atlantic. And that water is being forced upward along the shelf break, and eventually some of it makes it over. And in this particular plot is showing it getting right to Grays Reef. This really explained what we were seeing with these peaks, because this water, this high CO2 water, is being pushed up along the seafloor, does not come to the surface, but being pushed up along the seafloor, and that's exactly why we're seeing these peaks of higher CO2 on our seafloor platform and not up there on the buoy. In this particular one, you can see green is the sea surface CO2 levels and blue is the seafloor CO2. So it's not reflective in the buoy, and this is a classic case of where you really need to have your instrumentation down on the level of the organisms you're wanting to monitor. If we were solely doing by what we had seen in the buoy, we get a very good overall concept of what is happening at Grays Reef as far as changes in superficial seawater chemistry, but we would be missing an important part that during these higher levels of CO2 that are coming in on the seafloor, these organisms have to be stressed while this is happening. This is as CO2 goes up, pH goes down, these organisms have to be experiencing some level of stress. So this is a classic example of why these webinars that we're doing are very important, because I wasn't aware of the data that Rick had done until he showed it in his webinar. Okay, this is a really neat plot of seawater CO2 versus pH that we did at Grays Reef. Now this is a completely natural phenomenon that's happening there that is seasonal, where you see the CO2 increasing starting in January up into September, and the pH, how close it reflects. The higher CO2 reflects in lower pH. It's like a mirror image of the two. And what I like about this particular one is that it helps show people how tight that water chemistry is tied, that as CO2 increases, the pH is going to decrease. Now, in this particular frame that we're showing, it's about 7 months time period. CO2 changed by about 220 parts per million, and the pH changed about 0.23. So this is a fairly big change for organisms to have to experience within a short time frame. Overall, we're looking at the possibility of pH in the oceans changing by about 0.1 over the next century. So it shows that there are places where organisms have learned how to adapt to changing pH. Now we're also collecting water quality data out there. This is came from the seafloor. You can see how the change in salinity happened. This was basically when Georgia came out of its drought. We started getting a lot of rain. It actually reduced some of the salinity levels. You can see the major in blue, the major temperature change that occurs out there. And then the red is the pressure difference, which is showing us the tidal changes, the seasonal, the monthly tidal changes. And then dissolved oxygen down here on the bottom. This is actually in milliliters per liter. If you convert it to milligrams per liter, it would be more like about 7, and which is a perfectly acceptable level for organisms living on the seafloor. Now we're also doing turbidity and chlorophyll out there, and what I like about this plot is it shows us which one of these time periods of turbid water are from algal blooms versus, say, river input, when you have major amounts of river water coming out to the site, the more turbid river water. So it's able to give us a feeling for what is happening throughout the year. Now this data, the water quality data, has also been used by a number of graduate students in their projects, something that they would not normally be able to afford to put together. They're able to use this for their research. So I mentioned earlier that Georgia Southern had teamed up with us to try and decide what will possibly happen to the benthic community at Grays Reef as the CO2 increases and the pH decreases. So what we're looking at right now is starting with oculina, mostly because there's a fair amount known about oculina, and trying to find out what will happen as the pH decreases. How will oculina continue to thrive out there at Grays Reef? Now this is a fairly slow-moving effort because we're just using in-house funds between UGA and Georgia Southern, and hopefully we will be able to pick up speed and start getting a little greater funding level on here. So basically, why do we care what CO2 and pH measurements are at Grays Reef? Well, the higher the CO2 in the water, the lower the pH. That's a given. That's purely chemistry driven right there. And where that comes in is the lower the pH makes it harder for animals to grow their shells and skeletons, which means they're going to be stressed. Stressed animals do tend to have more problems. All right, the low pH, as I said, can stress the animals. It can impact the early life stage development. That's already been shown in a number of experiments that you don't have to have extremely low pH in order to do this. When you have the early life stages, you can get deformed organisms. They can have problems later in life. And it can affect reproduction and the overall health of the community. Well, where it really starts getting some attention is if it starts affecting the fish populations out there. So when people start going out to fish and they can't find any, and that's when they get upset. So the CO2 and pH measurements are important for the overall health and well-being of our benthic and water column community, that if we do not know what's going on and try and have some insight on what can we do to head this off, this is a very important topic, and we'll have discussions for many years to come. Now we also, of course, the most important part is to recognize those that are funding the efforts. And the surface mooring, as I said, is covered by funding from the NOAA Ocean Acidification Program and SECOORA. Our seafloor observatory is primarily the University of Georgia, and the benthic CO2 research that we're starting is UGA and Georgia Southern. But we couldn't get any of this done without the logistical support from Grays Reef National Marine Sanctuary. Without the vessels and the diver support, this program would not be able to happen. So we're very thankful to have them ready and willing to support this project. And so with that, if anyone has any questions, you can send them to Jen, and I'll do my best to answer them. And if I can't answer them right off, we can try and do that by email. So thank you, everybody, for listening.
Thank you, Scott, for a fantastic presentation. That was really informative and very exciting to hear you reference and be able to do what you did with Rick Wanikoff's information. That is just really, really exciting. So at this point, we'll open the floor for any questions for Scott. So if you have any questions, please type them into the question or comment box on the right of the GoToWebinar panel on your screen. So we do have a question, and let's see, it says, are the same PCO2 instruments used on the seafloor and other locations, and if so, have there been similar results or findings?
Okay, the instrumentation on the surface, like I said, the main driver on the CO2 is a LICOR analyzer, and it is actually measuring the gas. So it's a different method than what we have on the seafloor. The seafloor is doing dissolved gases that go through a membrane, and it actually is changing a reagent in color, and from there calculating what the CO2 are. So far, I haven't seen anyone. This is the only site that I'm aware of, at least on the East Coast, that is doing both surface and seafloor monitoring, and I haven't seen anyone else get the results of where they're seeing some of these CO2 spikes coming up from the upwelling. But there is I think PMEL had experimented with a few off on the West Coast. I haven't seen any results from that where they might be getting similar. Upwelling is much stronger on the Pacific coast than on the Atlantic, but so far this is the only one I've seen that has actually got PCO2 spikes on the seafloor that are not represented on the surface.
Okay, thanks. Let's see, there's another question here from Janine, and she wants to know, this is in reference to the Wanikoff presentation again, that slide, and she wants to know what process is causing that upwelling, and do you see a pattern for the spikes already related to season or some other situation?
As far as the driver, that would be best to ask Rick Wanikoff on that. I'm not exactly sure what the driver is. It doesn't appear to be seasonal because we've seen these spikes at various times during the year. Doesn't really line up with certain times of year, so that would be a good one to ask Rick Wanikoff on. If I tried to answer it, I'd probably get it wrong.
Okay. All right. And then another question is, what is the temperature record in relation to the elevated benthic PCO2?
The temperature, it does the same thing on the seafloor as it does on the surface. As the water cools down, you're going to see the PCO2 levels drop. And the same in the summer, as the water starts heating up, you're going to see PCO2 increase. Mm-hmm. So that part does work throughout the water column.
Okay, great. Thank you. I have a question for you, Scott. I noticed in one of the slides around that benthic sampler or instrument, Centropus striata, the black sea bass, which is not only a commercially important species but also a recreationally important species here, at least off South Carolina, I'm sure to some degree North Carolina and Georgia and Florida. So is anyone doing any OA work looking at any of the finfish, and in particular black sea bass, in response to OA?
That part I'm not really familiar with. You do not hear much about relating it to fish, largely because fish are more mobile. Typically, people pick things that are not mobile so that you can actually say that this organism was exposed to these levels for a certain period of time. With the fish, you don't know how much they're moving around so that they are more mobile. I do know that the black sea bass are very common out at Grays Reef. They seem to be doing well. They're actually one of the more curious when we're out there working. They have to be right there in the middle of things. But I'm not sure how many are actually doing finfish. That would probably be a better question for the NOAA Fisheries Group.
Okay. And then a follow-on question to that, you mentioned the oculina. I don't know if you were able to catch the Leslie Wilkes presentation looking at Lophelia, but that would be an interesting connection there, I think, to share information. So just a point.
Yeah, I think I missed that one. I believe I was out of town and haven't had a chance to go back and watch it, but since it is archived, I do plan on going back and checking that one out.
Are there any other questions from the audience at large? If not, I have one.
Yeah, Paula, we do have a few more. Scott, there's a question about Astrid is wondering if you're planning on looking at the plankton communities around the buoy at all.
At this point, no, and it's mostly just because funding. Since we are doing this in-house, we're starting very small. I see no reason why it couldn't be done, but that would have to take some kind of funding source in order to get out there and do that part of it.
And I did see another one from Steve Giddings. Scott, do you know if there are plans to expand the observing network for ocean acidification in the South Atlantic Bight or at larger scales?
There's still a lot of discussion on that, and actually through we're looking at trying to add whatever we can, and that comes straight down to funding. They're constantly wanting us to identify potential locations to add new buoys. But as I said, it comes down to funding. Whatever PMEL can expand to, they tend to get some extra money each year that can go into instrumentation. And as I said, it really comes down to funding.
Okay, thank you. And a question from Dennis. How often do scientists board the 20 buoys during the buoy's lifetime?
That's really hard to say. Last year I was out at the Grays Reef buoy, I think 6 times. One to do a complete change out of everything, and then the other times to do more minor repairs of replacing one instrument or checking on things. It's hard to say. Each buoy stays out there deployed for roughly 3 years, and then it gets changed out. So it's hard to say as far as during its lifetime how many times, but I do know that last year I was out there about 6 times.
Okay, thank you. And then from Susie, she wants to know if you're finding your SAMI CO2 and pH sensors to be reliable. And then and this is specific to the sensors, how often do you have to service them?
So far, both instruments seem to be doing fairly well. Typically, we try and recover them every 3 to 4 months to keep the fouling down. Once we bring them up, download the data, typically put in a new battery, clean them, and then they're set to go again. They usually have to go back to the factory for a complete overhaul about every 1 to 2 years.
Okay, thank you for that. And then Deborah wants to know, how does turbidity affect pH?
Turbidity doesn't really affect pH at all. That's more of a benthic community factor that we're looking at, turbidity really does not affect pH at all.
Okay, thank you. And then Ming-Sun wants to, it's a comment, it says if there's high PCO2, if the high PCO2 is due to upwelling, then normally you should see a significant drop in temperature. So that's just a comment.
I'll agree with that. We've looked at it, and same, you should see some change in salinity, which we haven't. Part of this, the changes are for fairly short time frame. They're not long, so I'm not sure, and this water is also mixing with existing water, so I'm not sure why we don't see a big drop in temperature as this water changes. But we do know that something is happening. I've also, to add to that, I've sent the raw data back to the factory when we first started seeing this and had them analyze it to make sure the instrument was not doing something strange, and they have come back several times and said the raw data looks good, and then it comes back down to match what we see in the buoy, which makes me realize it isn't instrument failure that's happening, but something that's actually happening on the seafloor.
Well, so it's been a long time since I've looked at a chart. Where's the Charleston bump in relation to Grays Reef?
I'm not sure exactly where that is. I don't have a chart in front of me, so it's kind of hard to describe where that is.
Mm-hmm. Yeah, I can't remember either. It's been a long time. So I'm just thinking about deflection of the stream, and I know that's surface, but still. Okay. I have a question for you. How much do those bottom instruments cost, just roughly?
To give them nice round figures, the seafloor CO2 unit is roughly $20,000. The pH is about the same, and the water quality unit is about $24,000, and that includes the long-term battery pack that goes with it. The battery pack is good for 6 months to a year deployment.
Okay, thank you. So if there aren't any other questions, if you think of any questions after this seminar, please email Scott at the email address that you'll see in just a little bit. So Scott, thank you again for this excellent presentation. And if you are interested in joining the SOCAN listserv and you have not already done so, please sign up at the URL listed on the slide that you see now. And please direct any questions about the SOCAN Acidification Network to SOCAN@SECOORA.org. We'd like to thank you for attending the SOCAN State of the Science webinar today. We welcome any feedback, further questions, or suggestions for topics about this webinar series. And you can submit any input on the way this webinar was administered today by replying to the following email that you will receive, the follow-up email that you will receive. And as mentioned at the beginning of this webinar, the presentation will be available on the SOCAN website along with the slides. So our next State of the Science discussion will be held in 2 weeks from today on Tuesday, June 30th. And Rue Morrison of the Northeast Regional Association of Coastal and Ocean Observing will discuss the development of the Northeastern Coastal Ocean Acidification Network at 12 p.m. Eastern Standard Time. So we hope that you'll join us. Scott, thanks again for a fantastic presentation, and to all of you for your thoughtful questions and taking the time to join us today. We hope to see you at SOCAN's next State of the Science conversation. This ends this presentation.